Howdy. Welcome back, everyone, from lunch. Hope you all are enjoying the symposium so far and getting most out of it. So next we have wonderful speaker, Dr. Corrie Moreau with us, and I have a great pleasure introducing her. Dr. Corrie Moreau is an ant biologist and professor of ecology and evolutionary biology at Cornell University and director of insect collection. Growing up in New Orleans, Dr. Moreau generated her interest in collecting bugs, which led her pursue undergraduate in entomology at San Francisco State University in 2000. She did her master's there in collaboration with California Academy of Sciences, which is a natural history museum in San Francisco and has world-class biological collections from every corner of the world. So she got her first exposure using that collection and got interested into studying ant evolution. And so she went on to Harvard University to get her PhD, and she had amazing mentors. Two, one of them was E.O. Wilson, and he's a godfather of ant biology. And then her another mentor was Naomi Pierce, who has done magnificent work in ant-butterfly interaction, who we were trying to get her for this symposium as well. But we have Corrie. Now, then I do want to point out how E.O. Wilson describes Corrie in his book, Letters to Young Scientists, which is a wonderful book. He goes on to say, there was no bravado in Corrie, no trace of overweening pride, no pretension. The story of Corrie Sox Morrow's ambitious undertaking is one I feel especially important to bring to you. So you can get more inspiration by reading Corrie's story in E.O. Wilson's book. Now, hopefully that gives you an idea about Corrie's tenacity and brilliance. Then, before going to Cornell, Dr. Moreau worked as a curator in Field Museum of Natural History, Chicago for 10 years, which is one of the largest field museums in the world. Dr. Moreau has published over 100 scientific papers in top-tier journals. She studies everything about ant evolution, whether it is their environment, their gut microbiome, what they are eating or interaction with other species, everything. She has studied ants across the world and has made tremendous contribution to the knowledge of ant evolution. There is a long list of awards and recognitions that Dr. Moreau has. I'll just point a few. She is a Cowley Fellow of National Academy of Sciences, Fellow of American Association for the Advancement of Science, and Royal Entomology Society. She is an advocate for increasing women and diversity in the sciences and has been featured by National Geographic as a Woman of Impact. In 2014, she was listed under 10 women scientists you should follow on Twitter. So make sure you're following her. Today, Dr. Moreau will talk about natural history, museum collections, insect science, and diversity in STEM, three critical aspects of ecology and evolution. And with that, please help me welcome Dr. Moreau to the stage.
Sorry, the evolution got cut off on the transition here from Mac to PC. So I want to thank all of you for being here today, and I really want to thank the organizers for coming and inviting me. I sort of feel like this is a joke. It's, an evolutionary biologist walks into an ecology conference. So I have to admit, when they invited me, I was like, me? I'm not an ecologist. But what I hope I'll show is that I actually do a lot of ecology in the sense of I try to incorporate everything there is to know about my group of organisms, whether it's their ecology, their behavior, phenotypic diversity, everything, and trying to understand them. But before I talk about my ant research, I want to talk about what I see are sort of three big challenges in biology and what the solutions are. And this slide actually came out because the National Science Foundation asked me to be a distinguished lecturer. And they said, we want you to sort of highlight what do you think are some of the big challenges. And I was like, oh, this is a chance for me to say what I think, like to the National Science Foundation. And I don't know if it'll have any impact at all, but I think that one that we have to think about, how do we sort of document and discover biodiversity? I know that we, in these days of climate change, we tend to think of like, how do we preserve the planet? But without knowing what's actually there, it's kind of, a futile effort. I believe that natural history museums are revolutionizing the way we think about science, and I'll talk you through some examples of that. And that we all must work to diversify science. And this is not just something that should land on the shoulders of a few. So why is it important that we discover and document biodiversity? I've been fortunate as a rainforest biologist to travel all around the globe. I've visited rainforests on every major continent. And it's really amazing to go deep into those lush jungles and sort of study your organisms in their natural habitats. But one of the things that we see across the globe is this. See, we're seeing this, translation of natural habitats into these human modified forms. And although what I'm showing you is in Uganda, that's happening right outside here at Texas A&M. It's happening outside the door of Cornell University, right? This is not something that's only happening in tropical biomes. We're seeing the conversion of these lands because human populations are growing so expansive. We need places to build our homes and to grow our food. Now, in these habitats, we often consider it much more dire because we have the opportunity to save these intact, very intricate habitats. Where for us, oftentimes we're like, well, if we convert our parking lot back into a wild, a meadow, maybe we don't have as much of an impact. So the reason I think natural history museums are revolutionizing science is that we're now using technologies to leverage them in ways we never thought possible. Clearly, these were the resources where people were doing taxonomy and to some amount, morphological systematics for hundreds of years. But really with the technologies available today, we can actually use them in much more diverse and important ways, I would argue. So of course, we're still using them to study anatomy, but in many cases, we're using technologies like micro CT scanning, right? So we can look at things in so much more depth and rigor than we were ever able to before. Of course, one of the things that's critical is all of that label information that's associated with every one of those specimens. Not only is it that we can sort of document where they were, we can look at how they're changing through time. Some of the strongest evidence for climate change comes from these species distribution maps and modeling, that the only way we can do anything with that is because we can see where organisms were and whether they're shifting their distributions or becoming extinct where they were. Of course, because of many of the now reduced representation genome sequencing methods and next generation sequencing, we can actually lever these specimens in a way that we never could before. Sanger sequencing required the ability to sequence huge fragments of DNA, where these actually require that on a fresh sample, we have to use a method to shear it into very small pieces to sequence it. Well, guess what? That's what degradation in museums do. So now we can actually take them and sequence either parts or whole genomes from specimens housed in these natural history collections. To some extent, we're starting to understand the microbiome of species and how those are changing through time through using museum collections. Less so for insect collections, because since all we do is stick a pin through them and let them decay, what we have is not their native microbiome, but sort of what's pervaded as they've gone through this decay process. But for many vertebrate groups, people have been cutting out stomachs and storing them in ethanol for a very long time, so we can actually go in and ask those kinds of questions. And that we're also seeing that many people are very interested in these ideas of nature-inspired design. In fact, this is a project that came out of the Cornell University insect collection by my collection manager and some faculty in material science. These material scientists were thinking about it one day and they said, how come when it rains and raindrops are so big, insects aren't all shattered and smashed to the ground? Like, how can a raindrop hit them and not crush them? So they came to us and said, could we just bounce lots of raindrops off of all of your insects in the collection? We said, sure. And what they were able to show is that the microfine structure on insects it's created in such a way that it fractionates the water in ways that we really weren't aware of before. So now these material scientists are trying to use that to sort of create new materials. And so I think what I'm trying to show to you is that natural history collections are foundational to almost all forms of science. But I'm going to take a moment and pause and say, you're all ecologists. And ecologists often don't interact with natural history collections as often as I think you should. There's two reasons why that I think you should. One of which, you're doing this beautiful work. Let's imagine you've gone out to this field and you've said, I'm going to study this group of plants or this group of insects. And I demonstrated that there's this one dominant species, and then there's these subordinate species, and here's how they interact through time, and you publish these beautiful papers. And then some jerky systemicist comes behind and says, you know what, I sequenced DNA from a whole bunch of these things, and that thing you thought was one, it turns out it's actually 3. So now how do we prescribe all that important ecological work you did to this correct species? If we can't go back to a voucher in a museum and say, oh, actually what you're doing is it's now called this, and that's how we're going to connect the scientific literature, we ultimately have to kind of ignore the work you've done because we don't know where to assign it. In addition, you guys are all working with the beautiful organisms out in the world, and I love ecology, and I've done this myself. This is not a shame moment, but we publish papers and we're like SPA1, SPA2, SPA-A, SPA-B. Well, that's fine because maybe you don't have the ability to identify it, but if you partner with a museum, they might be able to tell you, yes, that's a new species, or no, that's this species and it has this name. Having names on things lets us unite all that scientific literature. So hopefully you're all now are going to be much more engaged. You guys have beautiful collections across this campus. I happen to have visited the entomology collection. I know it's fantastic. I happen to know the curator of mammalogy and I know it's phenomenal. So please get in touch with your local curators. Now, the other thing is that I'll say is that, museums and specimens are clearly critical for scientific research across lots of fields. Obviously, as I've stated, documenting and describing populations, but also improving public health. We can't know where sort of emerging pathogens are coming from unless we can sort of find those source populations. We also know it's important for agricultural practices, right? We need to know if what's coming in is a potential pest or not. And that translates to food security. We also know that you can use museum collections to monitor things like environmental contamination. I'm a curator at the University of Michigan. Actually, I think it was when he was still a student at the Field Museum. He was able to go into all of these bird skins and demonstrate that once the Industrial Revolution came along, that we see a change on the metals that are found on the plumage of birds and that it's significantly changing through time. We can discover emerging pathogens, right? So we want to know where are the reservoirs for these things that are becoming problematic, not just for us, but for our livestock or for our natural wild populations. And ultimately, we need to think about how these can be leveraged for things like conservation of endangered and threatened species. And ultimately, as I mentioned before, we can get these bio-inspired designs. So really, these often serve as that fundamental material for understanding things like climate change. So are animals and plants shifting their distributions? Are they going locally extinct? I would also argue that we all have to actively work, and I don't care what your identity is or how much privilege you hold or don't hold, we all have to actively work to ensure that our workforce mirrors our populations. And so the reason that it's so important to have people engaged in STEM from all sort of backgrounds is that we know that there's not currently enough trained PhD candidates in STEM fields. Now, I know that people often say, well, there's not a lot of academic jobs. Yeah, that's true. Guess what? Being an academic is just one of many diverse career options for people with PhDs. And we need PhDs in many fields across biology writ large. We also know that when you have diverse teams, you have increased innovation, creates creativity and problem solving, increased teamwork, and you're more likely to not only attract great talent, but retain great talent. Now, you don't have to trust me. There's a whole body of literature out there. These are just 2 examples of it, showing that when you have diverse work teams, you have higher profile and higher quality science, but also has higher impact. And I would actually argue that museums and sort of increasing diversity in STEM can go hand in hand. We know that museums are often one of the first forward-facing places that people who often aren't actively invited into science have an opportunity to sort of learn their place. But in addition, as we digitize these collections, we're making them accessible to people around the world. All right, so now I'll sort of put back on my insect evolutionary biologist hat and talk about sort of why I'm so interested in sort of these kinds of questions. So ultimately, what I want to understand is what are the processes that generate that beautiful biodiversity when I'm sitting in the middle of the rainforest, right? I'm an ant biologist, that's why I'm there, but I can't ignore the screaming peahas and the stinging plants with the massive thorns that you learn quickly in the tropics. When you fall, you never reach out and grab a plant, you just hit the ground because the plants are meaner than the animals. And so I try to think about what are the abiotic and biotic factors that are sort of structuring this diversity through time. So you think about precipitation, humidity, rainfall, soil salinity, right? These are things that are impacting every group of organisms found there. Even if it's not directly, it might be indirectly through the food web. But I also like to think about these biotic interactions. So there's lots of things that are shaping the organism's interactions through time, predator-prey interactions, and all kinds of symbiotic interactions, from things that are incredibly parasitic, which we know there's probably more parasites than anything else in the world, through things that are mutualistic and are engaging in these, in many cases, obligately reciprocal relationships. And so really, I want to think about those things. But as an evolutionary biologist, I see the entire world through the lens of a phylogenetic tree. That's literally what I see. And I think that what's so powerful about this is it tells us a lot about the organisms we're looking at. It doesn't just tell us who they're most closely related to, and although that is very important, I'm sure as ecologists, you've often been told, like, well, you need to control for phylogeny, which, seems like this pesky, statistical test you have to do. But it's important to know whether something's evolved once or whether it's evolved independently. But there's many things once you have a phylogeny you can do, and that's where I get excited. So as an entomologist, this tree is really boring to me, right? But what I love about a phylogeny is that I can then ask the evolution of traits. I can say, how many times did those spines evolve? How did they shift their distributions through time? What was their migration pattern across Gondwana, right? And I can only do that through the lens of a phylogeny. In addition, I might want to understand, why did something sort of diverge from each other? Why did we have a speciation event? Is there something we can sort of explain through time? I mean, the Hawaiian archipelago is a beautiful example, but we can think of other things like maybe we had an aridification event that dried up to, very, moist habitats. We had a mountain range sort of, arise, and then it was one widespread population. And then through time, through drift, and maybe some amount of selection, we now have different species on those different barriers. Almost all the work I do is all based on DNA and genomes. And so what I'm often trying to understand is, why do we have differential rates of molecular evolution across the phylogeny? Why is it that some clades have really fast rates of molecular evolution? Why do we see sort of increased rates of molecular evolution happening independently, consistently? Is there something about the biology of that organism that explains that? And then ultimately, if we have some kind of external information, maybe it's the fossil record or an important geologic event, we can put a time frame on that and ask, when did those special characters arise, right? And did that sort of explain that sort of increase in diversification? What happened? Was there some kind of geographic, explanation for why that speciation event occurred? And what's the time frame for those shifts in molecular evolution? So one of my PhD advisors was E.O. Wilson, and he once said that ants are the little things that run the world. And I really bought into this, because ants engage in symbiosis across the tree of life. They're a really great set of organisms to use if you're interested in symbiosis. We know that they engage in these animal interactions, right? So this is a scale insect that produces that little drop of honeydew. In return, the ant will aggressively defend it from parasitoids and predators. We know that they engage in symbiosis with lots of kind of plants. This is an obligate ant-plant symbiosis system that I've actually done some research on. I won't present today, but what we found is differential rates of molecular evolution across mutualists compared to generalists. We know that they engage in symbiosis with lots of kinds of fungi, and sort of the quintessential example of that is the leaf cutting ants, right? Or the fungus growing ants. But they're not all of these interactions are positive. So this is the zombie fungus, Orthocordyceps, right? Which manipulates the behavior of the ant host so that it's beneficial for its own reproduction. And so I've collaborated with David Hughes's lab to do some work on the evolution of cordyceps, Orthocordyceps in ants. But more and more, my work has sort of turned more inward looking inside the ant and thinking about their microbiome and all of the bacteria and fungi and microscopic eukaryotes, and how that might be shaping the evolutionary history and ecology of these hosts. And so the further I get into this, I sort of think that E.O. Wilson sort of had it right. I now say that the microbes are the little things that control the little things that run the world. And in fact, I think that's probably true for all of us. The question is, did we really decide to eat pizza and salad, or did our gut microbiome tell us to eat that pizza and salad? All right, so the first thing I want to do is highlight some work we did, actually leveraging museum collections to ask a question that could not be addressed any other way. Oh, it turns out that I didn't put that in there. That was that missing slide. I'm so sorry. I opened this up and there was an entirely blank slide, which means all of my things went away. So I will tell you about it. So what we did is we went into, so the Xerces blue butterfly is this icon of insect conservation. So it went extinct over 100 years ago. It was found only in the coastal sand dunes of San Francisco. And it was believed to have gone extinct. Well, we know it went extinct because of human encroachment and conversion of its habitat. But the question was it a true species? Because there's another closely related species that has a distribution from Canada throughout almost all of California down into the northern part of Mexico. And a lot of people have speculated like it was just an isolated population of this widespread thing. And so, this is sort of, the icon of insect conservation. The Xerces Society is named after this exact butterfly. And so we thought, well, we can actually address that question now with these new sequencing technologies. And so we went in and took a specimen that had been collected in the 1940s, cut off part of its abdomen and ground it up. Well, we didn't really have to ground it up, it was almost powder anyways. And we didn't have to do any of that sort of shredding the genome because it was already shredded. So fast forward through lots of sequencing, what we were able to demonstrate is that the Xerces blue butterfly was not only a distinct species, its closest relatives were not even the things that had been predicted to be closely related to. And so what that shows is that this species was in fact driven to extinction. And this has big implications because there's several efforts now to sort of de-extinct the Xerces blue. Now, the reason for me that that's troubling, and I love insects, I'd love to have more of them, is that we actually know that it was engaged in obligate interactions with certain plants that are no longer found in the Bay Area. So now we have to do plant restoration. We know that it engaged in symbiotic interactions with native ants that tended it, and the introduction of the Argentine ants are potentially one of the last sort of straws that broke the camel back on why this thing disappeared. So how are we going to eradicate those and reintroduce the native species if we're going to do this? Now, I know George Church is on a big bend to sort of, stop to sort of, re-extinct a whole bunch of things. And I say that that's actually a waste of our time and our money. When we have things that are hovering on the cusp of extinction now, we should be putting those resources towards that. So I'm sorry that I don't have the slides. It's a published paper. I think it came out in 2021. You can read it. All right. So now talking about ant-plant interactions and how they may have increased their interdependence through time. So anybody who's studied plants sort of in a paleo environment would sort of agree with this slide, which is that plants were pretty diverse until more recently. And now we're sort of living in the age of angiosperms. So almost every plant we see is a flowering plant now, right? And with that burst in diversification, lots of other groups sort of took advantage of these new niches. So it won't be surprising to know that many insects that feed on plants or pollinate plants went through massive bursts in diversification. But it wasn't just things that were feeding on the plants. Things like these epiphytes and many ferns are also thought to have gone through massive diversifications because the low light of the canopy provided an ideal habitat for them to flourish. And in fact, the same thing is true for ants. We also see shifts in diversification of the ants correlated with the expansion of the angiosperm forests. Now, what we really wanted to try to understand is, well, how does this sort of explain this shift in diets? So the ground plan for ants is that they're predatory, they're eating other arthropods, but we also now see that many ants have shifted their diet onto these flowering plant resources. So they're feeding directly on plants, through things like these extrafloral nectaries. These are produced by the plant to attract ants to serve as their sort of outsourced army. That many plants that are engaged in symbioses also create these food bodies that they feed to their larvae. That some plants actually produce this seed coating called an elaiosome specifically to attract ants to have them to disperse their seeds for them. And then many ants actually feed indirectly on plants through the mediator of a sap-sucking insect. And so ultimately they're eating only plant-based resources while guarding their army of sort of their herd of cattle. So what we want to try to understand is sort of how did this association evolve between these two groups of organisms? Are there sort of ages that align when we see these different traits arising? And is there some sequence by which they evolved? So the first thing that we did is we took a very large plant phylogeny, but we wanted to incorporate the fact that many ferns actually also have extrafloral nectaries. So we added a whole bunch of ferns to this phylogeny. And then we went in and did some ancestral state reconstruction on traits we think that are important for their interactions with ants. So these are exactly the things you saw before. So extrafloral nectaries, elaisomes, and these special structures called domatia. And these are where plants produce specialized structures for the defensive arthropods to live 100% of their time on. So they now no longer live even on the canopy floor. We then went in and inferred a very large phylogeny for the ants. So this is the largest data-driven phylogeny for ants still to date. It has over 1,700 species in it. And this is mostly spearheaded by a former postdoc in my lab, Matt Nelson. And here again, we went in and did ancestral state reconstruction to try to understand some of those associations with plants. When are they taking advantage of plant-based resources? So I'll sort of walk you through the overall findings of this paper. So I want to remind you that the ground plan for ants is that they're nesting in the ground, they're foraging in the ground, and they're eating other arthropods, right? They're predatory. And if we look at when these particular traits arise in this interaction between the ants and plants through time, so along this x-axis, you're looking at the present through 150 million years ago. And what we see is the first thing that happens is that ants begin to forage arborally. So they're no longer looking for food resources only on the ground. Next, we see that they begin to include plants in their diet. So they're up in the canopy, they're finding plant wounds, they're finding, things that they, nectar they can feed on, and they're taking advantage of that food resource. Next, we have the plants start to recognize that there's this army that when you have ants on your plant, whether they're actually going to aggressively defend the plant or not, herbivores are less likely to feed on that particular plant. We see a response from the plants and they evolve extra flow nectaries. Now we see ants begin to nest arborally. So, right, they're no longer even coming back down to the forest floor. They're living and feeding entirely in the canopy. Now we see the plants have evolved eliosomes, and these are incredibly important structures. They've evolved over 100 times across the flowering plants and over 12,000 species. And now we have the plants produce the most sort of sophisticated structure that's only for holding their defensive symbionts. And so what we were able to show is that ants were utilizing plants long before plants evolved these specialized structures, which is exactly what you would expect. We see this incremental sequence of evolution, right? It's not bursts. It's not sort of, these things all happen at once. And so if we think about the timeline of ants, we see that they went from feeding and nesting and foraging on the ground to then taking advantage of the fact that they had this, additional axis of space that they could forage for food, where they could build their nests. And in between, the plants are sort of responding to the fact that they can reliably use the resources and army produced by these ants to their own benefit. Now, it wasn't something we actually set out to test in this study, but it was something that had been sort of, spoken but never written down. And that was that, when you have ants that are predatory, for them to get to the herbivorous stage, they have to transition through this generalist stage. And what we're able to demonstrate is not only is that in fact true, but that once ants become herbivorous, we see almost no reversions ever to even being a generalist and no reversions to being a predator. So it seems that once you get locked into this highly specific diet, you're sort of there for life. But I can share that many ant lineages actually went through bursts in diversification because now there's no longer competition, there's open niches and resources. So for a long time, I've thought about ant-plant interactions and how ants have sort of transitioned. Herbivory has evolved independently across the ants dozens and dozens of times. And, through time, I started thinking about, well, okay, if you have these, organisms that once fed entirely on animals, now are eating entirely plant-based diets, how are they actually doing that? And there had been some, really nice early work that had sort of hypothesized that maybe they were using these symbionts, but we wanted to understand if that could in fact be true. So we wanted to understand whether they were facilitating the evolution of herbivory across ants. And so, as you can probably tell, I think ants are the best system to ask almost any question. But I really do think they're a great system to understand things like gut bacteria. And that's because we can do these really amazing things. Since herbivory has evolved independently many times, we can say, do we see the same mechanisms happening, the same changes to gut structure? Do we see the same bacterial communities? But in addition, we can actually manipulate their diet in real time. We've actually gone out and done that. We change their diets and watch how their microbial communities respond. Because they're living in these socially interacting colonies, we can split them. They're almost genetically identical and then do sort of controls as well as manipulations. In addition, since they're socially interacting, we can ask questions of transmission, both through evolutionary time, when are they co-evolving with particular lineages, but also what happens if you introduce a pathogen to an ant nest? How is it sort of mediated so that it limits sort of harming the queen or the brood? We also might have expectations that we wouldn't expect every bacteria that we find in the gut to co-evolve with the host. So can we make predictions about when we'd expect to see that versus not? And also, how are they sort of sharing genes through time? And then lastly, because microbes are everywhere and ants are almost everywhere, except Antarctica, we can ask the question, when do we see, the acquisition of these microbes? Is it from the local community? Is it something that's shared through evolutionary time? Do closely interacting ants more likely, share microbes? So when I started this work with my longtime collaborator, Jacob Russell, who's faculty at Drexel University, I sort of joked that when he and I first started this project, I feel like we were those early rainforest explorers like Humboldt or Bates or Wallace or Darwin. Literally, we like got to the rainforest and we're like, look at all of this stuff. And we just started grabbing one of everything, right? And that's really what was happening when we were looking at the gut communities of these ants, because very little work had been done prior to us. So the first thing we did is we took almost 400 individual ants from almost 150 ant genera and just asked what's there. Now, that's a very descriptive project, but necessary. And what we found is not only a lot of diversity, but what really jumped out to us is that this group of bacteria was really in high abundance across the ants. And this is the Rhizobiales. And for any of you who work on plants, you're probably familiar with another group of bacteria from this order, and that's the Rhizobia. So rhizobia are these bacteria that are found in the root nodules of leguminous plants. We know that the plants produce these specialized structures to house the bacteria and give them carbohydrates. And in return, these bacteria fix atmospheric nitrogen and provide it to their plant hosts. So why are we finding a group of bacteria that's related to this within the gut of ants? Are they doing something similar? Now, another reason hint we have that this might be important is it has to do with sort of the trophic ecology or the food web of ants. So what you're looking at here is the stable isotopic signature of ants from things that are highly predatory like army ants, all the way through things that are entirely herbivorous. And the way that we do this is we use the ratio of heavy to light nitrogen as it accumulates up the food web. We always have known predators and known herbivores and plant material as the base for these food webs, but what I'm showing you is just the ant data. And each of those diamonds is an ant genus where we have multiple samples to get their isotopic signature, if you will. And what you'll know plotted on this y-axis is association with that group of bacteria, Rhizobiales. And we only see ants associating with Rhizobiales that feed lower on the trophic scale, suggesting that maybe they are in fact enriching their diet somehow. But if you have a keen eye, you probably already noticed that there's two groups of ants that feed very low on the trophic scale and have no association with Rhizobiales. But these are the carpenter ants and their allies. And this is one of the few groups of ants that we knew anything about their bacterial communities. And in this case, they actually have a specialized structure, a bacteriocyte, that holds their symbiotic bacteria. And we know that they're actually providing nutritional resources to their ant host. So it appears if you're going to feed low on the trophic scale, you have to have an association with some group of bacteria. So what you're looking at here is a phylogeny of the bacteria, but color-coded and labeled by the host or environment from which it came. And everything in red is from an ant. Now, almost all the samples grouped together in what we call this Rhizobiales specific clade. And interestingly, these are mostly either herbivores or generalists that often eat plants. The only samples that we have that are actually outside of this are known predators. So we can't rule out that they haven't eaten something, clearly not an ant, but something else that also has bacteria in this group related to it. So we essentially have this one acquisition of this group of Rhizobiales within the ant hosts. And within that, what's even more interesting is that the ants that are more closely related to each other are grouping together. So these are the bacterial sequences, right? So then you might ask the question, well, what happens if we look on the flip side from the ant perspective? And there what we see is we have at least 5 independent associations of this group of bacteria with ant hosts. We did some statistical tests and we were able to demonstrate that there's a strong correlation with being herbivorous and in many cases also being arboreal and having associations with these groups of ants. And some people have speculated that these, rainforest canopies are essentially food deserts. There's lots of plant material, but not a lot of other resources. So these ants are capitalizing on those plant-based resources and likely using bacteria to supplement their diets. Now, if these bacteria are in fact supplementing their host diets, then we'd expect to see some amount of tissue specificity. So what we wanted to understand is what does the bacterial community look like in each of these specific parts of the digestive tract? So just to remind you, the crop is the social stomach. It's just a holding chamber. It's where workers, when they're out in the environment, they can swallow liquid food, they can come back and regurgitate it to other individuals. They in fact have a very tight filter here. That's this thing here called the proventriculus that makes it impossible for things just to flow through unless it's purposefully released into the next segments of the digestive tract. Next, we have the midgut. We know that's where digestion begins. And then we have the hindgut, which we broke out into the large intestine, also called the ileum, and then the rectum, which where we know is many things are getting ready to be excreted. So we've gone in and done these careful dissections of each of these digestive tracts for multiple individuals within a species across many species of turtle ants. And this is work that was really spearheaded by a current graduate student in my lab, Peter Flynn, who'll be graduating this May. He's done a lot of beautiful work. This is actually the minority of what he does. Mostly he works on viruses. So keep your eyes peeled to learn all about the viromic community of ants. But what you'll see is that when we look at the crop, remember, no digestion occurs there. It's very diverse, but each of these bands, that's an individual, ant digestive compartment. We see that there's not a lot of diversity within a sample, lots of diversity across samples. Once we move into the midgut, where we know some digestion occurs, it's almost entirely dominated by one group of bacteria, the Opotutales. Once we move into the ileum and the rectum, those bacterial communities look more similar to each other, but they're really pretty diverse and pretty stable across every single sample. And that's, of course, sort of played out here in this NMDS plot. Now, this thing, the gaster, that's essentially the abdomen, because doing these careful dissections where we pull out each specific compartment, these ants are tiny. They're like smaller than a grain of rice. It's really hard work. We wanted to know, can we as a proxy just grind up the abdomen and get the same answer? And the answer is yes, so that's great. So we don't have to always do these careful dissections. But it's not just the diversity of bacteria that's different. It's also the abundance. So what you're looking at here is the overall abundance across these, or in looking at every single sample. So even though the crop was really diverse, there's almost nothing there. It's just contamination on whatever they ate last. Now, once we move into the midgut, those communities are both, very abundant and stable, right? In that case, it was almost all Opotutales. And the midgut also is mirroring quite a high abundance. Again, once we get to the rectum, we think that that's just sort of the dead cells that are being excreted out. So this is another line of evidence that these bacteria are likely enriching their host's diet. Now, if you remember back, I told you that the rhizobiales, that specific clade, was related to the rhizobia that fix atmospheric nitrogen. So we wanted to know, could that be what these gut communities are doing? So we used two methods to look at this. One, bacteria that fix atmospheric nitrogen have specific genes called NIF genes, and that's the genes that let them convert that, those atmospheric ions into usable things for a host. So we went in to see if we could find those genes. We could not find full functional genes of the NIF genes. We also went one step further and did acetylene reduction assays across both many different species of turtle ants. We did single ants, we did, cohorts of ants. And in all cases, we see no signature of the fixation of atmospheric nitrogen. So if they're enriching their host ant's diet, it's not by this means. Another indirect line of evidence we have comes from this really nice fluorescent in situ hybridization image where we've lit up the bacteria. And this is work that was done by my colleague, Peter Lukasik. What you're seeing here is this is the midgut. So the crop is off screen. This is the midgut. This is the ileum, or that large intestine. And this is the rectum. And you can see the rectal pads there. These spaghetti-like structures are called Malpighian tubules. And so for any of you who haven't taken insect physiology for a while, I'll just remind you that the Malpighian tubules are essentially the kidney of the insect. They're just loose in the body. They're finding uric acid waste. They're depositing it into the digestive tract so it can be excreted. But one of the things we know is that that is a great building block for, building, especially for essentially any of the amino acids, but especially the essential amino acids. So if bacteria are going to use this resource, they're actually in their highest density right where the Malpighian tubules are dumping that uric acid waste. Now, we do have some direct lines of evidence. The first comes through both genomic sequencing of the bacteria within the gut, but also metagenomic sequencing, since many of these bacteria are almost impossible to get in culture. My longtime collaborator, John Wirtz, has been really, sort of at the forefront of culturing many of these. It turns out not only are there many, and we have to culture them, but there's also many strains. And so it's still ongoing work. But we have been able to sequence the genomes and metagenomes of these. And what we've been able to demonstrate is that they have the full complement, the bacterial genomes, to synthesize essential amino acids. So they can produce these for the host. Now, the question is, are they providing them to the host? Because it could be that they're synthesizing them either for their own use or their interactions with other bacteria in this community. So we went in both with my collaborator, Jake Russell, but also my collaborator, Yi Hu, and we did a bunch of manipulative experiments. In this case, what we did is we used antibiotics to cure and knock down the bacteria in some cohorts. Others, we left them intact. Some, and then we fed, stable isotopes, enriched isotopes to some, and some we did not. So what you're seeing at here is when we left the bacteria intact, but didn't give them the labeled urea, compared to when we gave them the labeled urea and knocked out their bacterial signatures. For the essential amino acids, we really see no difference between them. But when we leave the bacterial communities intact and give them the labeled urea, suddenly we see a huge enrichment of those bacteria in the ant host. So we didn't sample these from within the gut tissue. We actually sampled it in the free-flowing hemolymph in the leg. And we're able to demonstrate that these are being transmitted from the bacteria out of the gut lining into the tissue of the host. And in this case, instead of sort of the fixation of atmospheric nitrogen, what's in fact happening is that they're recycling nitrogen and converting that into amino acids for their hosts. So now we sort of know that these gut bacteria are important to their ant hosts, but we wanted to go one step further. Well, how is the host using them? And I wanted to understand if this could be important for cuticle formation. And so the way to think about an insect's cuticle is as its armor, right? It's sort of what protects it from its outside environment, not only from sort of abrasions and injury, but it's also a great way to lessen desiccation. And what we also know is that across the ants, there's a lot of variation in sort of how we see them building this armor. And I will share with you that the reason this project ever came to be is that I have spent countless hours dissecting out thousands of individual ant guts. It's tedious work. I can't have coffee. And if you know me, I live on coffee. So that's not an ideal situation. But as I've sat there breaking open thousands of ants' cuticles to get to those gut compartments, I noticed something. I never set out to do it. It wasn't a hypothesis I had beforehand. It was one of those scratch your head moments. Like at some point I was like, I think I'm seeing something. So I kept looking. And what I saw is the cuticles that were the hardest to break through. They were either predatory or they were herbivorous, and we knew they had gut bacteria. Many ants have a very flimsy cuticle. It's easy to tear through. So I thought, could it be that the bacteria are helping them build that really tough armor? So just to remind you about the structure of the cuticle. So the cuticle is primarily made of chitin, which is a biopolymer. Within that biopolymer, we can have cuticular proteins embedded in it. Now, in order for the cuticle to sort of become hardened, or we call it the tanning process. So the tanning process, I'm sorry, I'm going to take you down a little bit of a chemistry pathway. And I promise you I'm not a chemist, so I won't go too deep in the weeds. But the tanning process starts with phenylalanine and tyrosine. And from there, it goes to DOPA, and it can go down one of two pathways, either the melanization pathway or the sclerotization pathway. The way to think about it is the melanization pathway is the sort of darkening of the cuticle, where the sclerotization pathway is more the toughening of the cuticle. So the insect cuticle is made of chitin, as I've already told you before. But in order for the chitin to grab onto anything, it requires a linker. And the thing that it usually wants to grab onto are those cuticular proteins that are embedded within the cuticle. But for the protein to attach to the cuticle, it requires the imidazole arm of histidine. Now, there could also be other embedded particles like melanin, right? That's the color particles. We also know that there can be metals embedded. I'm not going to present it today, but we've done a study where we've been able to demonstrate that there are differential levels of metals embedded across the cuticle of ants. We've now looked at many species, but there's no metals deposited within the sort of abdomen or thorax, whatever an ants we call it, the mesosoma. Sorry, we all have to have different language. But and there's no metal deposited there, but we see an enrichment of metals on the tip of the mandibles, right? So it decreases wear. We also see it on the tips of the tarsal claws, but we don't see it in the sting, even in ants that are highly predatory and using their sting. And that's surprising because an ichneumon wasp that use their sting to burrow through wood, they actually do deposit metals. So that's pretty interesting. All right, so back to the cuticle. So this is a really nice paper that was published in 2015 by people I don't know. But what I really liked about it is it's showing how that tanning process works on the mandible, on the mandible of a grasshopper. So I guess Hojin's and his lab are happy to see this slide. And what we see is that sort of tanning process through time. This is a composite image. And what we're seeing is from the fully untanned material all the way through the fully tanned material. And what they were able to demonstrate is what you see is that the amount of oxygen within that material stays constant. What we see is an increase in the nitrogen that's sort of displacing the amount of carbon during the tanning process. And in addition, what we see is that we see an increase in the sclerotization tissue and proteins, but the amount of chitin more or less stays the same. And that's really what leads to toughness, hardness, or strength. If there's any material scientists in the room, I apologize. It turns out each one of those terms means something very different, and I never can remember which one. We're actually working with the material scientists now to understand this, but I apologize now. I don't know the difference. So going back to our turtle ant system, I already told you that they have bacteria in their gut. We know that can take urea and uric acid, translate those into phenylalanine and tyrosine. So the question is that in fact what they're doing? And is that how they build that tough cuticle? So it turns out we weren't the first ones to think about the contribution of symbiotic bacteria to the formation of the cuticle. There's been exactly 5 studies prior to ours, all done in beetles. And what they were able to demonstrate is that when you give antibiotics to these beetles that have obligate symbiotic bacteria, that they decrease in color, right? So they become depigmented. And we see that the cuticle thickness decreases significantly. Now, in only one of those papers could they hypothesize a pathway. So they actually had the genome of the symbiont, and they could show that the symbiont had all of the steps to synthesize the essential amino acids minus the last step. So they posited that the host must do this last step. So they have a proposed biosynthetic pathway, but they actually are not confident in it. So what we wanted to know is which specific cuticular components are involved. I already told you that it's not just melanin color, right, and thickness. So we wanted to know, is it the proteins, the linkers, the chitin, or the melanin? Are there other potential pathways? Are we just going to recover the same exact pathway that they found in a beetle? And are these mechanisms conserved across many species? I'm only going to show it to you for the turtle ants today, but the question is, every time we see these herbivores evolve with these symbionts, are they sort of doing the same thing? So what we did is took lots of colonies of this ant. This is my favorite ant in the world, Cephalotes varians. It's the Florida turtle ant. It's amazing. I could talk about it all day. But what we did is we took these colonies, we split them in 2. Some we didn't give antibiotics to, some we gave antibiotics to. Then we gave an enriched urea to all of them again, so that we could trace these isotopes through this experiment. Because the adults already had formed their cuticle, we needed these components to go into the larva and go through all developmental stages, right? So that we were measuring things that had been in the manipulation. So we had to run these experiments for a long period of time. We marked all the adults that were present at the beginning of the experiment so we could then rule those out in the further downstream analyses. And that's just to remind me that anytime you're seeing a purple nitrogen, that means we enriched for it. We then did some careful gut dissections to confirm whether we were seeing what we saw in the previous reports. And we used liquid state NMR to examine those. We then scraped out all the insides and had only the cuticle left, ground those up, and used solid state NMR to look at both the enriched nitrogen, but also at C13. And just to remind you, if you haven't taken chemistry in a while, NMR is just an analytical chemistry tool that allows you to follow a single isotope within a sample. We also used scanning electron microscopy to see whether we saw changes in the anatomy or structure of that cuticle. So the first thing we did, and this is that's liquid state NMR, where we're looking at here whether we see differences in the amount of tyrosine or phenylalanine within these gut tissues. And of course, what we see is when we give them antibiotics, we see significantly less of these components. So this was sort of a check to make sure that we were manipulating the system in the same way we had previously. We also looked at whether we saw a shift in C13, and you remember we did not label for this. So this is also a nice sort of confirmation that we hadn't messed up the system, that when we look at those individuals that had gone through the antibiotic treatment and not, there's no difference in their sort of chemical shift within the NMR analysis. Now, we wanted to understand what we should expect to see for this. So it turns out you can buy whole giant jars of powder of chitin, just pure chitin from your favorite chemical warehouse. And we use that to sort of pinpoint where our standard should be. So like you saw here, there's like 1 million peaks. We don't know which one's chitin. So in order to figure that out, we had pure chitin. So that tells us exactly where it should be. We then went in and took some of these ants that we had done nothing to. They were straight from nature. And we looked at what would happen if we looked at their cuticle. And what you see is that we see that same shift peak, right where we see the chitin. But we also see that the peak is much wider. And I told you before that it has other things embedded in it, like cuticular proteins. So we'd expect to see that. You'll also notice that our spectra is much lower because unlike the giant jar that we pulled off the shelf and just scooped stuff into the machine, here we had to take tons of ants, scrape out all their insides, grind up their cuticle. So the amount of starting material is so much lower that we're always going to see lower peaks. All right, so here's what happens when we give them antibiotics. So anytime we see a blip, it means that that's the contribution by the ant host itself, right? Because they can't use their bacteria. So we are seeing a little peak right around where the chitin is. So we know that the ants are contributing to that. And again, just to remind you that when they're enriched from the bacterial, they'll be in pink versus in black. That means they're from the host. And here what we see is there is a little bit of a blip. And we aren't those in this case are probably from the ant host itself. We don't really understand the mechanism yet, but we also know that they're contributed by the bacteria. And this is the guanine arm of arginine. That's the thing that allows the protein to attach to the chitin. Now, if we look at when we include those samples in which we left their bacterial communities intact, you can see they're significantly different. We see a huge enrichment of this nitrogen within the cuticle. We again see that peak that corresponds to the chitin in the embedded proteins. We also see the guanine arm of histidine has a blip, in one case stronger than the other. We also see that the cross-linkers are involved and that the imidazole arm of histidine is involved. But interestingly, in the imidazole arm of histidine, you might have noticed that this is a cyclical structure, but only one of the nitrogens is coded. And that's because there's some work that was done in the 80s that demonstrated in insect cuticles that we should, for the imidazole arm of histidine, we should see 3 blips, and we only see 2 of the 3. And that last nitrogen is actually provided by the host very far along in the biosynthetic pathway. Now, when we use that sort of scanning electron microscopy to look at the thickness, we see exactly what they saw in the beetle study. So these are not to scale, but this is exactly half the width of the other, suggesting that the bacteria are in fact contributing not only to sort of the cuticular components, but also the thickness, the amount they can produce. But unlike in the beetle studies, we actually see no change in color. And I actually knew we would see this because we've been doing antibiotic experiments on our ant hosts for a very long time to manipulate their bacterial communities. And I never saw any hints of depigmentation. So it appears that it's affecting that tanning pathway, but not the melanization pathway. And so what this allowed us to do was to sort of not only show that we have a different biosynthetic pathway, but that we sort of have this iterative process where the ant comes in at key points and contributes. And that we see that the bacteria actually contribute to the proteins, the linker, and the chitin, but not the melanin. So hopefully what I've been able to convince you is that using sort of an iterative approach and diverse tools, you can start to really tease apart questions, not just in evolution, but also in ecology. Specifically today, I hope I've been able to convince you that museum collections are really critical to all kinds of scientific data and questions, that we have to all work to make science more diverse and inclusive, that in this case, the ant-plant interactions evolve through this sort of increasing interdependence, and that symbiotic bacteria facilitated ants to shift onto these herbivorous diets. And it does beg the question of sort of the chicken or the egg. Like when you shift onto the diet, what we know from the first study is once you become herbivorous, it's pretty difficult to transition back. So who's in control? The bacteria that they're now reliant on, or is it the ant host, right? And then, specifically, how are they contributing? In this case, we know that they're helping them build this tougher armor or cuticle through having this symbiotic relationship. Now, they're likely using the contributions of these bacteria in other ways as well, but this was a very direct way we could measure the contributions of the bacteria into the phenotype of the host. So with that, I'd like to acknowledge many people. Specifically, I'd really like to thank all of the amazing collaborators I've had through the years. I really want to take a moment and thank the members of my lab. I get to work with a really diverse, incredibly intelligent group of people every day. I'm grateful also that we're no longer only seeing each other through Zoom boxes. And of course, I want to thank my funding sources. And if there's time for questions, I'd be happy to take any.
Questions?
Yes. Was it too much ants or too much evolution for you guys?
Yes, Richard.
Have you looked for fungi in the ant guts?
Yeah, we have looked for fungi. It turns out it's really boring. And part of the problem is what we've looked at. So I've sort of told you this beautiful story, but all from the adult ant perspective. And so ants have this, many Hymenopteran do have this anatomy where as adults, there's a really tight constriction in their waist. So many of them can't eat solid food items. So ants as adults don't eat solid food. That's why they're always running around to nectaries and things like that. And the reason they're carrying these giant dead items back to the nest for the larva to feed on is the larva look like a fly maggot. It's just a sac with a mouth. They also need a lot more protein. And so we've looked at, it turns out we've been missing a lot, not just with the fungi, but also with bacteria, and that we actually expect to see a lot more diversity of fungi and different bacteria. So we've looked a little bit at the bacteria. So in adults, the fungi are boring. In the larva, we've just started looking and it looks a lot more interesting. So we'll have to definitely follow up with that more.
No, there's a question.
Thanks, Corey. That's a great talk. So I really appreciate what you're saying about the value of museum collections. But I have a little bit of experience. A few years ago, I tried to do a study where I needed to reach out to museums, and I did. And a lot of my emails went into black holes. It was really difficult to get responses. It was hard to figure out what museums had which specimens. And I'm just wondering,
Yeah,
I mean, it was a while ago. I don't know if that's changed. I know a lot of museums are trying to change that, but I wonder if you could talk about a little bit about developments in that area.
That's a very loaded and packed question. So I'm going to parse out the things that I think are appropriate for this audience because I could go on for hours. The problem is that for entomological collections, by and large, they are significantly the largest piece of any museum. So I was at the Field Museum. The Field Museum has, we brag, 30 million specimens who's ever counted. 17 million of those were just the insects. The next most diverse group was botany with 3 million. So like, we're just dealing with this massive amount, right? I'm at the Cornell University insect collection now. It's a tiny collection, only 7 million specimens. Have I looked at all 7 million of those? No, definitely not. So to be able to database those and make that information freely available, it's very laborious. If you've ever looked at insect pins, you know there's like 500 labels stacked together, all pressed like this. So what that means is that we have to take every label off, we have to transcribe it, we have to put every label back on, and then a unique barcode, move to the next specimen. And that's without data, like taking any digital pictures of the insect itself. So it's just going to take a long time. Now, to your thing about reaching out and not getting a lot of responses, I think we're seeing a sort of fundamental shift in how people think about museum collections and what curators' roles are through time. And I don't think that it's better or worse. I think it's the culture that's been driving it. We've all seen it, and you'll hear the horror stories if you talk to anybody who's an old professor, is that, oh, we used to have this massive collection, and then the administration decided just to throw it in the dumpster or to give it to some other collection because it was too much work to keep it up. So I think historically curators felt like they had to be very protective. It was like they didn't let anybody in because they were afraid if they let anybody in, somebody would be like, it's not used enough, or I could use this space for something great. So if you keep everybody out and tell everybody that you're important, it's harder for people to justify getting rid of you. Now with all these digitization efforts and sort of National Science Foundation and the National Academies just released a report 2 years ago about the importance of these collections, and that we are the baseline data for almost every climate study now. I think people are changing sort of the administrators, how they think about these collections, which then lets the curators become more welcoming. So I will just tell any of you, I only work in insects, but I know curators at lots of, and you guys have some in the room. If you're running in these kind of hurdles, sometimes it's just, you don't know who to talk to, right? You sent an email to somebody who's like an adjunct curator who comes in once every 6 months and they don't check their email, right? So sometimes it's just putting you in touch with the right person because it can be hard to figure it out. So I want you guys to all use collections. If you want to redo that study, reach out to me. I don't have to be an author. I'll just make the connections.
Thank you.
Hi, Corey. Over here.
Oh, hi.
Good to see you. Wonderful talk. So there's one part in there that had me absolutely puzzled. That was the abundance of the wide microbiome in the crop. Do you have any hypotheses how that could come with it? So they're just effective in cleaning their crop out, but it looks weird.
Yeah, I think what it is that, so an ant goes out, it looks for a food resource, it finds that one and sucks it up till it literally can't hold anymore and walks straight back to the nest, where then it distributes it. So anytime we're sampling an ant that has a crop, it's literally got just one food resource in it. So I think it's just the contamination on what it drank. Like it drank from this plant where this other ant drank from this plant, or this ant's found in the Amazon and this plant's found in the Chicoan forest. So the microbes are just going to be different, right? But I do think that, I don't think there's much exchange happening there, but it does make you wonder if there's some mechanism for keeping it depauperate. The other thing I've wondered is that, so now we have the mechanism for them to seed their guts is that as adults, they have a very narrow window. So you remember I told you that the cuticle kind of goes through this hardening thing. You saw that in the grasshopper mandible. Well, that proventriculus, the valve in the gut between the crop and the rest of the digestive tract, is also made of cuticle. And it's pliable and sort of soft enough that there's a 3-day window that an adult ant has to acquire its full component of its gut microbiome to be healthy. And if it doesn't do it within those 3 days, the gut's not seeded. But what that means is that all the bacteria pass through that, the crop, which it's not clearly seeding. They all pass through the midgut. Only one thing's staying, and then everything else gets stuck everywhere else. So not only is it how do they keep the crop sort of clean, but also how do you keep the midgut with only one group of bacteria? And this is going to be way too deep, I'm sorry, but bacteria battle with each other with these biosynthetic gene clusters. We happen to know that from the genomes that the bacteria within the midgut have none of these bacterial gene clusters. So they're not battling the other ones. I think it's host control. I think it's pH or the amount of CO2, and it might be the same mechanism by which they're keeping the crop clean. Great question, though. We haven't got that far yet. It's coming.
Thank you.
Hi. So I thought it was really interesting what you brought up about the lack of metals in the stingers. Are you planning on studying like if that's specific to ants, what the reason is, or you're like across hymenopterans?
Yeah, there's a little work done in hymenopterans, but it's very few, in fact. And I don't know people who are doing much work on that. I think it would be super fun. It's not going to be me. I have to say I love wasps, but wasps sting too much. I don't really want to get them. They're mean. I'll stick to my ants. If you want to do that, sure, people would love to see that work.
All right, I think, yeah, with that, let's thank Corrie.
Thank you all.